Welcome to the virtual lecture on Chapter 9. In this video, we're going to cover genetic mutations and Mendelian genetics. The first thing we're going to do is we're going to watch a friendly overview of Mendelian genetics.
These days, scientists know how you inherit characteristics from your parents. They're able to calculate probabilities of having a specific trait or getting a genetic disease according to the information they have from the parents and the family history. But how is this possible? To understand how traits pass from one living being to its descendants, we need to go back in time to the 19th century and a man named Gregor Mendel. Mendel was an Austrian monk and biologist who loved to work with plants. By breeding the pea plants he was growing in the monastery's garden, he discovered the principles that rule heredity. In one of the most classic examples, Mendel combined a purebred yellow-seeded plant with a purebred green-seeded plant, and he got only yellow seeds. He called the yellow color trait the dominant one, because it was expressed in all the new seeds. Then he let the new yellow-seeded hybrid plants self-fertilize, and in this second generation he got both yellow and green seeds, which meant that the green trait had been hidden by the dominant yellow. He called this hidden trait the recessive trait. From those results, Mendel inferred that each trait depends on a pair of factors, one of them coming from the mother, and the other from the father. Now we know that these factors are called alleles, and represent the different variations of a gene. Depending on which type of allele Mendel found in each seed, we can have what we call a homozygous pea, where both alleles are identical, and what we call a heterozygous pea, when the two alleles are different. This combination of alleles is known as genotype, and its result, being yellow or green, is called phenotype. To clearly visualize how alleles are distributed amongst descendants, we can use a diagram called the Punnett square. You just place the different alleles on both axes, and then you figure out the possible combinations. Let's look at Mendel's peas, for example. Let's write the dominant yellow allele as an uppercase Y, and the recessive green allele as a lowercase y. The uppercase y always overpowers its lowercase friend, so the only time you get green babies is if you have two lowercase y's. In Mendel's first generation, the yellow homozygous pea mom will give each pea kid a yellow dominant allele, and the green homozygous pea dad will give a green recessive allele, so all the pea kids will be yellow heterozygous. Then, in the second generation, where the two heterozygous kids marry, their babies could have any of the three possible genotypes, showing the two possible phenotypes in a 3-to-1 proportion. But even peas have a lot of characteristics. For example, besides being yellow or green, peas may be round or wrinkled. So we could have all these possible combinations: round yellow peas, round green peas, wrinkled yellow peas, and wrinkled green peas. To calculate the proportions for each genotype and phenotype, we can use a Punnett square too. Of course, this will make it a little more complex. And lots of things are more complicated than peas, like, say, people. These days, scientists know a lot more about genetics and heredity, and there are many other ways in which some characteristics are inherited, but it all started with Mendel and his peas.
Okay, so before we jump into genetics itself, we need to talk more about genetic mutations, which are actually the source of all variation and traits. So we've talked a bit about genetic mutations in our previous lectures, but it's basically any change or alteration to the sequence of a gene. Now we're going to expand on that definition by saying it's any alteration to the wild type nucleotide sequence of a gene. So what is the wild type version of a gene? Well, as we're going to learn here in Chapter 9, for every gene out there, there's usually multiple versions of it. Sometimes way more than just two versions, sometimes hundreds of various versions. But the version of a gene that's most common in a population living in a certain geographical region. Now, for humans, we're a global population, so that's harder to visualize, but if you're talking in the wild, okay, whichever version of a gene is the most prevalent, that's the wild type version. So we want to ask ourselves, why is one version of a gene more common in a population than other versions? And that really hits on evolution and how harsh the environment is in the wild. So the reason the wild type version of a gene is the most common is because that gene gives organisms what we call a selective advantage, meaning it allows them to survive and reproduce at higher rates than individuals that have other alternate versions of that gene. So it's advantageous to have in a biological sense, meaning you're more likely to survive and you're more likely to produce kids. So you're more likely to pass on that gene, which is why it becomes over generations more common in the population. But I do want to point out that just because something is advantageous in one region doesn't mean it's advantageous somewhere else. Okay, so better is a relative word depending on what your environment looks like around you. Doesn't necessarily mean faster or smarter or bigger. It just means better fitted, better suited to survive a particular habitat. All right, so let's go to the next slide. I also want to reiterate that genetic mutations aren't intentional, they're accidents, they're mistakes that can happen in various ways. We've already seen they can be errors during, DNA replication, mistakes by enzymes like DNA polymerase, maybe the mistake evades your repair system, maybe you came into contact with a carcinogenic molecule or UV light. But in any case, these can happen, these mistakes, these changes to nucleotides can happen anywhere in your genome, any of your 6 billion letters, A, G, C's, and T's. So they are random. But you need to realize that these random mistakes, it's almost like trial and error, okay? Every once in a while, you could produce a new gene, not necessarily a new gene, but a new version of a gene that will give you an even better protein that will give you a selective advantage. So it's going to give you a new protein and a new trait. But it's these mutations which have a negative connotation to them because, well, usually they are bad, that sometimes create new genes, new traits that are better for the population. So I have this little side note that says, think about humans and think about eye color as it's a character. So it says the wild type gene for eye color in humans is the version that results in brown eyes, and that's true. It's the most common in human populations. So then I want you to ask yourself this: where, do other eye colors come from, like blue or gray, like light colored eyes? How did they arise? And the answer is, at some point, you can actually trace this in modern biology. You can go back and trace mutations, but it had to arise from a genetic mutation. So mutations create variation. And so, as I've been saying, there isn't, just one version of a gene in a population. Sometimes there's multiple slightly different versions. So we have a proper name for a slightly alternative, different version of a gene, and that term is allele. Okay? So any various versions of a gene are considered alleles of one another. So the wild type allele is the most common version of a gene. So what, distinguishes one allele from another? Just their sequence. Sometimes it can just be one letter difference. Sometimes it can be a few, okay? But it's always a difference in their sequence of A, G, C's, and T's. So you might ask yourself, what do we call the other varieties of a gene that are not the wild type version? Well, in biology, we call those the mutant versions or mutant alleles. Not necessarily bad, again, just not the most common. Okay, so any other version that's not as common as the wild type is a mutant allele. And again, many genes have multiple alleles that exist within the population. Some are very rare, but there's oftentimes many out there. So just like I said, mutant allele doesn't necessarily mean bad. They usually have no effect or a negative effect on the health of the organism, but there is always the potential for a mutant allele to become the new wild type allele if it conveys some type of advantage in a certain time and place. And that's really how evolution works. So on the side, I say, think about this. The wild type version of a gene is around because it works best in a given environment. It's really a result of harsh sculpting by the brutal pressures of the environment over hundreds of thousands to millions of years, many, many generations. Okay? And I say harsh because all the other varieties, they're not there because they're not surviving and they're not reproducing. So the wild in the environment is very harsh and it picks off what doesn't work. That's evolution. By natural selection, that is. And I'm going to take you through 2 examples to understand these new terms. We're going to understand the terms allele, wild-type allele, mutant allele, and selective advantage. So my first example is embedded into my PowerPoint slides, and I just kind of made up situation here. So it says, shown here is a population of sea bass. This particular population living in this environment is comprised of around 950 bass with brown scales and around 50 fish with blue scales. They specifically live in a relatively shallow marine environment where the background color of the sand is dark brown. So you might, see that might be a good thing, like to camouflage with the sand in terms of survival, right? So I also say, suppose that a single gene is responsible for the coloration of these fish, the color of their scales. We will denote the version of a gene, the allele that codes for brown scales, as B. So I'm going to write that down. It's hard to write on PowerPoint, that's why it's a little sloppy. But and we'll also use lowercase b for blue scales. Okay, so 2 different versions of a gene. One version we're going to denote is uppercase B. That's really a nice trick in biology. We don't want to write out like 2,000 nucleotides. Let's just call it a single letter, right, for short. So uppercase B and lowercase b. And then more importantly, ask you a few questions. Which allele is the wild type version of the gene? How do you know that it's the wild type? Well, which version is most common? And that would be the allele that codes for brown scales, right? Most of the fish, 950 out of 1,000, are brown. So we're going to say uppercase B. How do we know this? It's the most common in the population. Now, there might be other subspecies or other versions of this fish that live in different areas, and maybe in those different areas, brown isn't the best to have, but here it is. And then lastly, what do we call the other alleles that are not the wild type, aka this blue version? Well, in this specific example, that would be the mutant version of the gene, the mutant allele. All right. So to further reinforce these concepts, we're going to bring in some harsh selective pressures, okay, to understand why, in this case, brown is the wild type. So I'm going to read this a little bit. It says, every environment, every unique habitat on the planet exerts its own unique multitude of what we call selective pressures. Now, you need to know that a selective pressure is basically any factor in the environment that basically kills organisms, picks and chooses who lives and dies, okay? So there's a lot of things that do that in the wild. Things mainly like predators, bacterial infections, viral infections, competition for food, water, shelter, the climate, the temperature, the humidity, the precipitation. All of these things affect whether an organism is going to survive, reach reproductive maturity, and have healthy, fertile offspring. Okay, so whatever genes, whatever alleles equal that survival, well, those individuals are going to stick around, and their alleles are going to become common because they work in that environment. So here we added something. We added a selective pressure, specifically a predator. So you can see the dolphin coming in from the right. And we're going to say that the dolphin is a visual predator. Okay, so it can distinguish between the brown scales and the blue scales. And the blue scales are really out of place because they stand out, and the dolphin can see them more easily. So the dolphin is going to pick off and eat and kill the blue-scaled fish at a higher rate than the brown. And that is why this allele is more rare in this environment, because of a harsh selective pressure. So lastly, let's restate which allele is the wild-type allele. That would be uppercase B, coding for brown scales. Why does it confer a selective advantage? Well, like I just said, it allows those brown-scaled fish to camouflage, giving them higher rates of survival and reproduction. And I have one more example that I made up for you to understand these terms and concepts. What I'm doing here is I'm combining really Chapter 9, which is genetics, and Chapter 13, which we're not going to be able to get to, unfortunately, but I'm covering all the core principles of evolution with genetics. And that really those two concepts are tied together. Evolution just looks at genetics over generations for the whole population. So it works, it fits. But here's my next example. I say in this example, suppose that in a certain species of rattlesnakes, there's a single gene, which we're going to call gene V, and that gene is responsible for a toxic protein in the venom of the rattlesnake. So we're going to pretend the gene's short. Obviously, genes are not this short, but we're going to say it's only like 7 nucleotides long. I've written it here in green. So we're going to say, here's the nucleotide sequence of the gene for rattlesnake venom. And that's the gene that works. Okay, it works because the venom is going to allow the rattlesnake to poison, right, their prey, make it easier to eat their prey, more food, better survival, better success. So this is the wild type, it works. It's a good toxic protein. It's doing its job for the rattlesnake. But what if one member of the population has a random genetic mutation? Now, again, it could make the toxin better. Maybe it doubles it, makes it more, But in this case, we're going to say it's a detrimental mutation. So we're going to say, oops, that T right there accidentally got changed to a G. So we're just going to substitute incorrectly 1 nucleotide. And we're going to say that has a pretty negative effect. We're going to say that completely messes with the information needed to build this toxic protein. Okay? So because of this mutation, we're now going to have basically a toxin, a protein in this case, that has a completely different wrong shape, and it's no longer a toxin. Okay? And that's not a good thing for our snake. Now it has the inability to kill its prey with a toxin. So you might wonder, well, why does one letter completely change your whole protein, right? Aren't there a lot of words and codons? Changing one letter shouldn't be a big deal. Well, we haven't really got to that. I'm saving that also for Chapter 9 here. The various types of mutations you can get and their various effects on a protein. So we're going to go over that moving forward. So this would now be, right, this would be an allele, this new version. And we would call this a mutant allele. You wouldn't expect it to stick around at a high frequency in the population. That's not very advantageous to have. And this would be, just going to put WT for wild type. The wild type version. And one more time, let's write out what a selective advantage is. So individuals that have this version of the gene for the toxin, we expect them to have 2 things. Higher rates of survival. They're getting more food in this case. Better defense. That's 1. But there's 2 parts to saying selective advantage. Number 2, usually it implies if you're surviving, better chance at reproduction and passing on your alleles, your genes, to future generations. All right, so that summarizes this whole section on genetic mutations. So I'm going to get us ready for our next picture. And now we're going to move into actual, what we call Mendelian genetics. I will actually define that term, Mendelian genetics. It doesn't just mean that it has to do with Gregor Mendel. It means something else. We'll define that at the very end. So first, I want to say a little bit about Gregor Mendel. He was doing these experiments that we're going to cover in the late 1600s, around 1859 to 1865. And he's from current-day Czech Republic, which was part of the Austrian Empire, studied at the University of Vienna, grew up as a farmer, pretty poor. He was really bright and smart, and ended up basically being both a monk and a botanist. He was really well-rounded at a monastery, which at that time was the primary kind of institution for science, education, and the church at the same time. And so he was doing these experiments in the abbey, or the garden of the church, pretty much. So since he's interesting, I think, and I'm going to talk about him quite a bit here in my last chapter, I want to show you a video giving you more background than I just gave you on Mendel.
Welcome back to Nature League. When most people think about DNA and the history of genetics, the names Watson and Crick usually come to mind. Or perhaps Dr. Rosalind Franklin, who provided the photo that helped the duo figure out the structure of DNA. But before the idea of DNA even existed, there were scientists who were studying the inheritance of traits despite not knowing the molecule that makes it all happen. One such scientist is Gregor Mendel, and he's considered the father of modern genetics. If you've heard his name, you might have learned that he was a monk, messing around with pea plants. While this is true, there's so much more to the story. Ah yes, the story of Gregor Mendel, the father of modern genetics. Let's take a look at this story. Johann Gregor Mendel was born in 1822 in Heisendorf, which was located in what is now the Czech Republic. He was the middle child of two sisters, and his family were farmers. He worked on the family farm when he was young, which likely fostered a love of nature, the outdoors, and plants. Yes, we're purposefully foreshadowing here, just roll with it. Johann was smart. Like, really smart. So smart that people took notice and urged him to leave the farm and go to secondary school when he was 11. Johann's father wanted him to stay on the farm because, labor and eventual inheritance, but his father also recognized the importance of education to escape the hardships of peasant life. Despite the financial strain, Johann attended two larger schools out of town and excelled, graduating with honors in 1840 when he was 18 years old. At 18, Mendel faced the understandable dilemma of figuring out what he wanted to do with his life. College? Gap year? Euro trip with buddies? Fast food joint? And by that I mean work on family farm? Mendel decided to continue his studies and enrolled at the Institute of Philosophy at Olmütz. Throughout these years, he struggled with both health and finances. As for the latter, his little sis came through big time and lent him some money from her dowry. Thanks, sis. As for his health, it set him back in his studies, so he struggled to complete the program. However, people were looking out for him and believed in his abilities. One of his college professors, Friedrich Franz, was a sort of scout for candidates for the Augustinian monastery in Brunn. Professor Franz recommended Mendel to these Augustinian monks, basically saying, look, the kid might be poor and kind of, sickly, but he's really smart, and you should give him a chance. Just do the right thing. At this point, you might be thinking, wait a second. If he joins a monastery, won't he just be monkeying around? Or where does education come in? Well, keep in mind that the monastery was a cultural center of that region during this time, and it was complete with a massive library and places for scientific experiments. The members of the monastery were composers, philosophers, and natural scientists, and science education was actually emphasized for those in the order. So it turns out the monastery was a pretty good fit for Johann Mendel. His parents agreed, and in 1843, at the age of 21, he joined the Augustinian order at St. Thomas's Abbey in Brunn. He took on the name Gregor from Gregorius. While there, his roles included priest, botanist, abbot, chaplain, and eventually a substitute teacher in 1849. It turns out Mendel was really good at teaching. He was like I'm really good at teaching. Students like me, other teachers like me, I'm gonna do this. And he went for a teaching certificate the next year. Despite his best efforts, Mendel failed the certificate exam. But one of the abbots saw potential in him and decided to ship him off to study at the University of Vienna. From 1851 to 1853, Mendel studied math, chemistry, and plant physiology. He returned to the monastery in Brunn in 1853 at the age of 31 and was put in charge of the gardens, which had been dedicated to agricultural experiments. He was still interested in getting his teaching certificate, but failed the exam again in 1856. So at this point, he was like, dang it, guess I'll dedicate myself to this cool experimental work I'm doing on the side. The experimental work Mendel had been doing centered around hybridization, which is the breeding of individuals from closely related species. He was interested in how traits were inherited. Keep in mind that at this time, the accepted theory of inheritance was blending inheritance. Basically, people thought that the traits of parents blended together to create the traits seen in offspring. However, in the Abbey's garden, Mendel saw that pea plants weren't following these blending rules, so he had to investigate what was going on. From 1856 to 1863, he crossed tens of thousands of pea plants with different physical characteristics to track these traits throughout several generations. The physical characteristics of pea plants he worked with were plant height, pod color, seed color, flower position, and flower color. By crossing plants with combinations of these traits, Mendel noticed certain mathematical patterns in the offspring. For example, when a plant with yellow seeds was bred with a plant with green seeds, all of the offspring were yellow. However, when he bred the next generation of those plants, only 75% of the offspring had yellow seeds and 25% had green. During this work, he made 4 very important observations. Number 1. Traits have multiple forms. For example, pea plants can be tall or short. Number 2, offspring get character traits from their parents through some kind of factor they inherit. I don't know what the factor is, but it's definitely happening. Number 3, in organisms with 2 parents, it seems like one factor comes from each parent through sex cells. Number 4, offspring outwardly express only one of the 2 factors they inherit from their parents. Let's say the factor that gets physically expressed is dominant, and the other factor is recessive. But Mendel wasn't done yet. He also noticed that when parents with differences in 2 traits were crossed, the offspring would have all possible physical combinations. For example, if one parent was tall with green seeds and the other parent was short with yellow seeds, the offspring were all 4 physical combinations. Since the offspring had combinations that were different from the parents, Mendel concluded that these traits were inherited independently of one another. Basically, he figured, there's no relationship between height and seed color. This observation was a big deal and is still taught and referred to as the law of independent assortment. Mendel was pretty pumped about his findings, and he presented them at meetings in 1865. He published the findings the next year, reporting that some invisible factors were providing visible traits in a way that was predictable. We now know that these invisible factors are genes, but DNA wasn't figured out until after Mendel had died. Most tales from this time that reference his work indicate that people sort of missed the point. Scientists misunderstood his findings and didn't recognize the significance at the time. He spent his later years too busy to focus on his experimental work and wrapped up in tax drama. Mendel died in 1884 at the age of 61. But was this the end of Mendel? Not quite. Though his ideas were largely unknown when he died, they were rediscovered some 15 years later. In 1900, 3 European biologists did their own experiments and came to the same conclusions as Mendel. Thus, the age of genetics was born, and in combination with Darwin's theory of natural selection and works by other scientists, the modern synthesis of evolutionary biology soon arrived. Although we've learned a lot since the time of Johann Gregor Mendel, his work still serves as a foundation for genetics research in the 21st century. His theories have fallen in and out of favor over time as new discoveries unfold, but his research is fundamental to understanding the field of genetics. Thus, he is considered the father of modern genetics. Thanks for joining us on this historical episode of Nature League. We've come a long way in certain fields of study, but it's always good to learn about how we got here. To keep learning about life on Earth, including the stories of those who have studied it, go to youtube.com/natureleague, subscribe.
Alright, so as you heard, Gregor Mendel is the father of modern genetics. Really the most interesting thing about Mendel is that he completed all of that work talking about these invisible and heritable factors that are passed from each parent before DNA was discovered, before genes were discovered, even before chromosomes were discovered. So that makes it really amazing. So as you saw in that video, before Mendel's work was accepted, the fundamental principles of genetics, the view is that all of the characters passed by 2 parents were blended perfectly in their offspring, like paint blending. We're going to see that that's not really the case. It can appear that way sometimes when you're looking at a character that's controlled by several genes, sometimes up to 100 genes, like skin color, hair form. But that's not really how it works. There's never, chromosomes don't blend together from your two parents. They stay separate, right? Homologous pairs. And also, if blending were true, you'd expect the whole population to look like clones of one another, right? Like a perfect blend of one another within a few generations. So that's not the case. So what we're going to do here first is I'm going to take you through two of Mendel's significant experiments. So I'm going to say we're going to jump into Mendel's garden, and we're going to recreate these experiments from his perspective. Then I'm going to go back and I'm going to relate these experiments to modern molecular biology terms, and I'll replace some of the things from this initial drawing with more modern terms. All right? So first we're going to look at it from his perspective. In doing so, we're going to define all of these terms: character, P generation, F1 and F2 generation, a phenotype, phenotypic ratio, the dominant trait, and the recessive trait. By the way, this is just a picture of a pea plant, which was obviously the model organism studied by Mendel. So our first definition is a character, and that's just any heritable feature that varies among individuals in a population. So let me start with humans, since that's a little bit more relatable. Well, you just did a genetics lab and you looked at 6 or 7 of your own human characters. For instance, eye color. Some weird ones too, right? Whether your tongue could fold or whether you could bend your thumb a certain way. But those are characters. Hair color is a character, skin color, height, okay? Like whether you can digest this sugar or not. There's a lot of characters at the cellular level that you can't see, okay? But any of those are characters. But he was working with pea plants, and he studied 7 major characters. Now we're only going to focus on one because the principles that he figured out from each character is the same, so it doesn't matter which one you pick. We're going to pick flower color, but he studied pea plant flower color, pea plant stem height, seed color, seed shape. All of these are examples of characters. Now a trait, also known as a phenotype, is a particular variety of a certain character. So since we're going to be talking about flower color, okay, 2 examples of traits would be, purple flowers or white flowers. Okay, those are particular varieties of a given character. Or if we're talking humans, let's say eye color. Eye color would be the character, brown eyes would be a trait, blue eyes would be a trait. So I told you he chose to study around 7 characters, and he specifically chose characters that only exist in this species of pea plants with 2 varieties or 2 traits, like round and wrinkled seeds, or green and yellow seeds, or purple flowers and white flowers. And to start his experiments, he always started with pea plants that were considered to be true breeding for these traits. So if we're talking flower color, he made sure that he had true breeding purple flowered pea plants and true breeding white flowered pea plants. Now what that means in a genetic sense is that if you were to plant the seeds of a true breeding purple flowered plant, that means all their offspring would be purple, then all of their offspring would be purple after that, the next generation. Okay, it's just purple. We'll have a better term for that when we move on in this chapter. That term is homozygous. And what he did is he took 2 different true breeding varieties and bred them together. So in genetics, a term for mating or breeding in an experiment is to cross two individuals. So he crossed true breeding purple flowered pea plants with true breeding white flowered pea plants. And when you do that with two different varieties, we call that a hybridization experiment. So their offspring are going to have, right, kind of like genes from the purple flower parent and genes from the other different white flower parent. So they're kind of like a hybrid of two traits. So we call those offspring that result the monohybrid offspring. So as I said, in the first experiment, he took true breeding purple pea plants and crossed them with true breeding white. So this original generation, our parents, the purple and the white parents, we call them the P generation, or the parental generation. As we said before, our specific character in this first experiment is flower color. The two traits are purple flowers or white flowers. So just to so this has an asterisk at the top, so it's not testable, but it's interesting. You've already learned in lab a bit about plant structure and physiology, so some of this will make sense. But this is kind of what Mendel did to ensure that it was a purple plant mating with a white plant. So basically he took a purple plant and cut off all the male parts, all the pollen-producing stamens, and just leaving that purple plant with the female pistil, the carpel. And then he took pollen from a white plant and dusted it onto the pistil of the purple plant. So he knew which plants were mating. Then he probably covered the purple plant so no other pollen could land on it. Then he allowed that the pistil to mature into a fruit. In this case, the fruit is a pea pod, and the seeds inside the fruit are peas. And then he grew up those seeds, so he planted the seeds, and then he looked at the phenotype or the traits in the next generation. So pea plants are a good organism to use, just like flies are and mice in certain plants like corn, Arabidopsis, tobacco. Why? Because they're really great for genetics because they have really short generation times. So you don't have to wait like 50 years to go from parent to offspring. You can do so in a few weeks. And they produce a lot of seeds. So if you're looking for a large sample size, that's great. So before I give away the results of his first experiment, I'm going to jump over here to my first drawing. And we're going to go through this together from Mendel's perspective. So we're going to fill in a few things. So first off, what do we call the original parental generation? They are the P generation. Here are the phenotypes, which we've said a few times, of the two parents. One's purple, one's white. What's our character? Our character is flower color. Now at this point, we don't know what the dominant trait is. We will soon find out. So to do so, I'm going to kind of uncover this. Let's see if it works. I'm going to use my eraser. So when he did this experiment, he grew up the seeds, the peas that resulted. Here's what the offspring looked like. Now there wasn't just one of them, there was hundreds of them, but all of their offspring were purple. Okay, so for the phenotype down here at the bottom, we're going to say they were 100%, all of them had purple flowers. And the last thing we want to fill in is, what is the name of this second generation? Well, it's called the filial one, filial meaning son in Latin. So we just call it the F1 generation. You can think of that as the first generation of offspring. Now, more importantly, think about this from Mendel's perspective. The status quo in terms of genetics and thought is that there's a blending principle going on, right? So you'd expect if you mated white with purple, you'd expect something like violet or light purple. And that's not what he saw. He saw that the offspring looked exactly like this parent. nothing like this parent. But that's a little confusing because obviously in sexual reproduction, this is passing eggs, right? Sperm, pollen. They're both passing something physical to their offspring. But whatever was being passed from this parent here, well, he was wondering, where did it go? You know? So his name for what was passed from each parent, he called it an invisible heritable factor because, again, he didn't know about genes. No one did. But that means gene pretty much. So the gene from the white-flowered parent, from his perspective, it disappeared. So he's wondering, where on earth did the factor from the white parent go? Did it get destroyed by the factor from the purple parent? Did it get pushed around and dominated? Is it hidden? What's going on? And we won't know the answer of that question until we get to experiment number 2, which is really a follow-up of this experiment. But at this point, we can fill in the last 2 parts of this drawing. This is where Mendel came up with the terms dominant versus recessive. So he deemed that the purple flower trait was the dominant trait. Why? Well, because in the presence of the white-flowered factor or trait, it seems that the purple wins out and the purple gets expressed, okay? The recessive trait in this case, white flowers, right? So when in the presence of the purple factor, so they're both passing a factor, this one here is not expressed, it's not showing up. So we call that the recessive trait. So we're now ready to go back. I want to make sure my voice is... Actually, I can't tell. Hopefully it is. Sometimes it can get turned off. But so these are the results. All of the offspring, all of the F1 plants were purple. And these are the questions he had. Why weren't they light purple? And then more importantly, what on earth happened to the factor passed from the white parent? Where did it go? This is from your book. We just went through experiment number 1 here, highlighted by the sun. Now, I'm going to do a Punnett square from Mendel's perspective. Okay? Now, we're going to do all kinds of Punnett squares here in Chapter 9. This will be our first introduction to one. Now, if you've ever done a Punnett square, you've probably used letters, uppercase and lowercase, instead of colors. But again, Mendel doesn't know anything about genes or alleles, so we're looking at this from his perspective. Then we'll go back and fill it in with modern terms. So from his perspective, you have the purple parent and the white parent. So the parents are on the sides. So you're probably wondering, how do they have 4 parents? Well, they don't. So let's start at the top here. These represent, as we're going to learn, homologous chromosomes, okay? So each individual has 2 factors, okay? So when they do meiosis, again, he doesn't know any of this, but when they do meiosis and they produce egg or sperm, there's 2 different, right, chromosomes they could give. Now, since it's true breeding, all they have is purple. So it doesn't matter which chromosome they give, the factor is always the purple gene, the purple factor. Same with the white, they're true breeding. So this is kind of what happened. Since they're true breeding, each parent gives a factor. So the purple gives a factor here. I'm going to fill this in the best I can. And basically what you're going to see here is no matter how the egg and sperm from the 2 parents combine, it's always going to look the same. They're always going to get 1 purple gene, 1 purple invisible factor from 1 parent, and a white factor from the other, right? They're monohybrids. They're a mixture of the 2 traits. Yet, even though these offspring have 1 of each, what did we say they looked like? 100% purple, right? So there was something about this factor that was allowing it to get expressed over the white factor. All right, so this will be more clear when we come back later, but we're going to keep it here because we're going to come back to it. At this point, we are ready for his second follow-up experiment. And I say follow-up because he took these purple monohybrid offspring and he allowed them to self-pollinate themselves and to cross-pollinate one another. Okay? So it's kind of weird, like the siblings pollinate each other, but again, these are plants, so it's a good genetics tool. So he did just that, purple crossed with purple. And so let's do that. So here are the two parents, purple and purple. Now this time, the original generation, this is still the F1, right? So these were those offspring. We wrote out their phenotypes. And what he saw, this is where it gets really interesting, was that their offspring were a mixture of traits. He saw that 75%, I'm going to write this down here actually, because I want to put it next to this part. The offspring are 75% purple on average. 25% of them are white flowered. So we can call this a 3-to-1 ratio. 3 dominant for every 1 recessive. And he did this experiment many times. And every time he got to the F2 generation, which this is, he saw this same ratio. 75% of them or so were purple, around 25% were white. So why is that striking from his perspective? Well, mainly because, wow, the white phenotype, the white trait came back. So it didn't get destroyed, it didn't get pushed around, it was hidden or masked, but it was there all along, it didn't get blended, right? Because 25% of these F2 offspring look, right here, just like their grandparent, right? So that factor was there all along in this generation, it was just hidden. So that further disproved blending. So next, we are going to do our second Punnett square. Now this represents the experiment that I just talked about. So we have our purple monohybrid parents on the sides. And this is where it will start to make sense why we have this 75-25, 3-to-1 phenotypic ratio, okay? And this is where Mendel really put it all together, okay? Because he was really good at math and statistics, counted a lot of seeds, and he came up with this repeating ratio and put it all together. So if we do this, so the way a Punnett square works, I'm going to go over them in more detail later, but basically you take, so this up here represents usually the mom on the top. So this is a possible egg. That's another way an egg could look. Both can't happen at the same time, but there's a 50% chance of each. Same here, here's the sperm. It could look this way half the time, could look that way, right? Homologous pair, we separate that homologous pair, it's like a coin toss, we pick one, could be this one or that one. We have to take into account all possibilities. And that's what this is, it's a statistical tool to figure out what the offspring of two parents could look like in terms of genetics. So it's a prediction. It doesn't tell you exactly what's going to happen, but what's likely to happen. So if we do this, the way it works is you go down and over like that. Here, down and over. Here, down and over. And that's how we fill it out, okay? So I'm going to do just that. So if we, number 1 here, go down and over, we'll need to highlight these purple. And 25% of the time, it's like getting heads twice pretty much. If you did 2 coin tosses, you could get a quarter of the time the purple factor from each parent. 50% of the time, you will get one of each, 1 purple, 1 white, or vice versa. 1 of each. And another 25% of the time, you'll get both of the white factors. Now, he figured this out. He said, wow, each parent must be passing on 1 factor, but each parent originally had 2. And the math made sense. He put it together to figure out that in order for the recessive trait to show up, you need 2 of the recessive factors so that there's no dominant purple factor to hide it. I'm going to put factors. Later on, we're going to call that allele, gene. But factors for now. Okay. So this will be our next one when we get there. So we're almost done with our second section here, genetics from Mendel's perspective. This is, really important results here. What he saw in the F2 generation, that 3-to-1 phenotypic ratio, 3 purple for every 1 white. And again, he repeated this thousands of times, and that ratio held true. Here's another perspective. This is exactly what he saw in his first experiment, 705 purple, 224 white. We just did this together. Now, just to kind of reiterate the main takeaways from this. Wow, the white flower phenotype came back. Wasn't destroyed, wasn't lost, wasn't blended. It was just hidden somehow. We'll talk about what's really going on because we know more about biology than we did in 1860. And again, this is where he came up with the terms dominant and recessive. To be recessive, you have to have 2 recessive alleles. To be dominant, well, you only need 1 of them and you're going to be purple, right? You could be purple-white, well, you're still going to be purple. You could have 2 purples, you're also going to be purple. All you need is 1 gene, 1 allele. And then, to say it again, since it's so important, what is that predicted phenotypic ratio that we always see when we cross monohybrids? 75% dominant, 25% recessive. Now, as we're going to learn here, this is not a pea plant thing. This holds true for all sexually reproducing organisms, any characters. So that would work for eye color, right? Anything where you have a dominant and a recessive version. This will hold true. And it held true for all of the other 7 characters that Mendel studied. He did this same experiment, starting with the P generation, true breeding, to the F1 to the F2, for 6 other traits. For instance, this one, where he saw yellow seeds were dominant to green seeds. So he crossed those 2, the true breeding parents, and he got all 100% yellow F1. And then like he did before, he crossed those F1 together, either through cross-pollination or self-pollination. Then in the second F2 generation, he saw 75% yellow, 25% green. You can even see his numbers that he kept track of right there. He did it a lot of times, but it always reduced to a 3-to-1 ratio. He was onto something. It wasn't just unique to pea plants or flower color. It might be universal to life. And now we're ready for section number 3, and we're going to cover some new terms here that really describe a lot of the same things we've already covered, just again from a more modern perspective. So we're going to go through those same 2 experiments and just add in a few things. So first, we actually covered this at the start of the lecture. It's nice to see it again. Many genes have several alternate versions out there in the population. Oftentimes there's more than one allele. Okay? Now, I didn't say this before, but a term for basically saying that a gene has a lot of different alleles, we say that there's a lot of genetic variation in the population for that gene, which is a good thing in terms of survival and evolution. It means you're more flexible to evolve if the environment changes. Variation is a really good thing, as we learn in chapter 8. The whole point of sexual reproduction and meiosis is to shuffle up all this variation and make as many different combinations as we can get. So variation's good. Now we're going to replace Mendel's term of invisible heritable factor. We're just going to say gene. So, relating to this flower color example, the gene for flower color is located at a particular spot on one of the pea plant's chromosomes, right? It has a locus, okay? The gene's at a spot. Now, the purple variety and the white flower variety, right? Those two different alleles, they're both located at the same spot on the chromosome. Their sequences are just slightly different from one another due to a mutation that happened at one point in time. But they're at the same spot, and they're intended to code for the same character, which is flower color. Now, for any basically plant, animal, or fungus, any sexually reproducing organism, again, all animals as well, you as an individual always have two alleles. You can't have more, you can't have less, because you have two parents. So for every one of your genes, you have two copies. One from your mom, one from your dad. Okay, kind of like one copy here, that's one. Second copy here from the other parent, two copies. Okay, so you always get two alleles. Now, some more important terms. Instead of talking again about factors, we're going to use alleles and genes. So our shorthand for alleles are always going to be single letters. One's going to be uppercase, one's going to be lowercase. So the dominant allele, which codes for the dominant trait, think like purple flowers, that's always uppercase. The allele that codes for the recessive trait, if we go back to our example with pea plants, white flowers, the allele that codes for that trait is lowercase. You don't have to, but the convention is to use the letter that is the first letter of the dominant trait. So if the dominant trait is purple, the convention is to use the letter P. If the dominant trait is brown eyes, the convention is to use uppercase B, lowercase b. Sometimes I'll use different letters because the uppercase and lowercase versions look too similar and it just gets confusing. Like, for instance, the letter C. So sometimes I'll change it up just for that reason. All right. Now, this is also pretty important. So I don't want you to confuse this with an earlier concept, because we've been talking a lot about alleles. And now you've heard four terms. You've heard wild type allele and mutant allele. And now I'm throwing two more terms at you: dominant allele, recessive allele. Are these different? Are they related? What's going on? Well, they mean different things. The dominant allele, all that means is for this allele, you only need one copy of it to exhibit the dominant trait. That does not necessarily mean that that dominant trait and that dominant allele is the most common. Okay? So dominant alleles are not always the wild type. You can have recessive alleles that are the most common. So don't confuse the two. They're not necessarily always the same. Now I'll say this, usually, more often than not, the dominant allele is the most common. We'll talk about why that is, but not always. Let me give you some kind of radical examples. In humans, having more than 6 toes or fingers, polydactylism, that trait of having more than 5, That's a dominant trait governed by a dominant allele. Not the most common, though. The recessive is to have 5. Dwarfism is caused by a dominant allele. Not the most common, though. So the recessive is most common. So, again, the dominant is not always the wild type. So, again, recessive doesn't mean bad, obviously. It could be the better version sometimes. So this is a really important slide. Remember I just said that as an individual, you have 2 parents. So for any given gene, you're going to have 2 varieties of it. They could be the same, they could be different alleles, but you only get 2, always. So when we're describing the 2 particular alleles that you have for any particular gene, that is your, as an individual, your genotype. That's just to say, what 2 alleles do you have? Now, in these simple examples here in Chapter 9, we're going to stick mostly with characters that are governed by a single gene where there's only 2 alleles out there. So that makes things easier. All right. So, since you can only have 2 alleles, there's only a few genotypes that you could be. And I'm going to write those out. Where do I want to write those out? First, I want to write them out here. Okay, so the first type is, what if you inherited for a certain gene? Let's pretend you're a pea plant. And let's say you got the dominant allele, the uppercase allele, from both parents. So you're big P, big P. What is that genotype called when you have 2 dominant alleles, both the same? Well, it has a name, and it's called homozygous recessive. Homozygous means the same, so you have 2 of the same alleles. Specifically, you have 2 of the same dominant alleles, so you're homozygous dominant. Sorry about it being a little more shaded. It's sensitive to pressure on PowerPoint, for whatever reason. What if, on the other hand, you inherited 2 lowercase recessive alleles, both the same but recessive? Well, we would say your genotype, the name of your genotype is homozygous recessive. Right? So you could have dominant, dominant, you could have recessive, recessive, or lastly, you could have one of each. You could have dominant from one parent, recessive from the other. Now, it could go from either parent. It could be dominant from one, recessive from the other, like this. But the convention is to always write the capital letter first. Okay, so we'll stick with this way. This has a name. That name basically means 2 different alleles, and that name is heterozygous. So if you have 1 dominant allele and 1 recessive allele at the same time, you're heterozygous. Now I want to tie all this in to our previous chapter to kind of tie everything together because it's all interconnected. We're just looking at this, basically the same concepts from different perspectives, different, first we were really zoomed in at the molecular level. Now we're kind of zoomed out more, but it's the same thing. So first we want to fill some things in. We have an uppercase P on the left. What do we call that? That's the dominant allele. Now sticking with our flower color example, this codes for the plant having purple flowers. The lowercase letter allele is the recessive. If you have 2 copies of it, you will have white flowers. All right, so the point of this picture here is to help you visualize what all this looks like on actual chromosomes, okay? So this is a real gene we're talking about, right? It's a long stretch of nucleotides somewhere along one of the pea plant's chromosomes. They have 7, by the way, 14 total. This is arbitrary, I'm just making it up. Let's just pretend this is chromosome number 1 of the pea plants. And we'll say the gene for flower color is located, well, where I put this line, bracketed by these lines right here. That's where it is on chromosome number 1. That's the locus, the location. I could pick anywhere, it doesn't matter. I don't know where it is. But how would this look like? So first we're going to say, well, what if the pea plant is homozygous dominant? Uppercase P, uppercase P. First, let's give it a name. So if you're uppercase P, it's going to have to be small. I need to fit it in here. You are homozygous dominant if you're uppercase P. Now, how would this look on the chromosome? It's going to look like this. So remember, this is a homologous pair. So this is like the chromosome from the dad. That's the one from the mom. Now, I want you to realize that a genotype is a shorthand way of depicting a homologous pair of chromosomes. Here's the letter, the chromosome from the mom. Here's the one from the dad, or vice versa. So the way this would actually appear in the chromosome is, well, we're saying from the dad, no, I want this one. From the dad, you're big P. Okay, that's one of the big P's. Now, the only reason you have 2 is because the chromosome's duplicated. Okay, but that's one of the big P's. And, hey, you know what? I'm going to change this up. Even though we're talking about flower color, just so this is easier to picture on my chromosomes, I'm going to pick 2 different letters. Okay? Again, letters are arbitrary, but I want you to be able to tell the difference between a P, an uppercase P, and a lowercase p. So I think the letter A would be easier. Okay, so same thing. We're just going to say the uppercase A is purple, lowercase a is white. So we're saying this individual is homozygous dominant. So from one parent, big A. And from the mom, they're also big A. So that's big A, big A. That's what that means. Okay, let's go on to our next genotype. So I'm going to erase this. What if the pea plant has one of each? What if they are heterozygous? Well, that is to say, from one parent, it could go either way. I'm going to pick this way. From one parent, they have big A. From their other parent, they're little a. Okay, so they're big A, little a. Now, I'll let you do homozygous recessive on your own. Okay, but that's what all this really means. Okay, it really relates to chromosomes. And we're moving along here. We're almost done with Mendelian genetics. We have 2 more pictures. Oops. So, back to the PowerPoint slides. We did that. That's what we just did. We showed how these genotypes would actually look on a pair of real homologous chromosomes. Just a more expanded definition of dominant versus recessive. Again, to be considered the dominant allele, like uppercase P, it just means that if you're an individual, a pea plant, and you have one dominant allele, all you need is one, you're going to be purple, right? You could be heterozygous or homozygous dominant. As long as you have at least one dominant allele, you're going to express the dominant trait. And likewise, the recessive allele, we call it the recessive allele because you need 2 copies of it to express the recessive trait. Okay, so that's the best true definition of dominant versus recessive. So now we are ready to go back to our original 2 experiments and get rid of those purple and white blobs and use actual alleles instead of invisible heritable factors. Now it should all really start to make more sense. Okay, so I'm going to do that. We're going to go back to first experiment number 1. Now remember, in this first experiment, he took true breeding purple pea plants and crossed them with true breeding white. So now we know a little bit more about what that true breeding means. Well, that probably means they're homozygous dominant, right? So we're going to put that up top here. We're going to put uppercase P, right? Uppercase P. They're big P, big P. The white parent, well, it must be white, right? The recessive trait, because it has 2 recessive alleles. It's little p, little p. Now, when you complete this Punnett square, right, when you go down and over, before I do that, I want to show you what you're simulating. Again, this ties into chapter 8 as well. The first thing you're simulating is a coin toss. You're saying there's a 50-50 chance when this parent, right, that's their genotype, when they do meiosis and they produce, in this case, eggs, they're going to split and separate that pair of homologous chromosomes, right? It could align either way during metaphase. You could get this one half of the time or this one half of the time. But we have to take into account both possibilities. In this case, it doesn't matter. They could line up either way. You're always going to get big P because they're both big P. But I want you to realize you're simulating meiosis, okay? So we'll do that. Now, it doesn't matter. For both, we're going to get 1 of each. The second thing you're simulating is random fertilization between the eggs of the one parent and the sperm of the other, right? The various ways they could combine. So, main point here, a Punnett square is a statistical simulation of meiosis and fertilization, right, sexual reproduction between 2 parents of known genotypes. And this is, we're looking to predict what could their offspring look like in terms of the principles of genetics. In this case, it's pretty straightforward. They're always going to get 1 big P, 1 little p, no matter how you line it up, right? Now that should start to make sense why, right, these were 100% purple. Because every one of them has 1 copy of the dominant allele. And that's all you need to be purple. Now, they still have the recessive, but it's getting masked, right? It's hidden by the dominant allele. So we can expand on that. We can say their phenotype, right? We already learned that. How they look, their appearance, 100% purple. We can also now say their genotype, 100%, what is it called when you have 1 of each? Heterozygous, 100% heterozygous. Hopefully that makes that a little more clear about why he saw those results. Now we're ready to do the same thing with experiment number 2. Now remember, experiment number 2 is a follow-up of the first one. So the parents are those same offspring from the first experiment. So they're the purple monohybrids. So they all have 1 dominant, 1 recessive. All right. Now this is where it should be more clear as to why we have to take into account when we separate this pair of chromosomes, you could get either, right? So half the time you could get big P, half the time you could get little p. So let's fill this out, right? Over, down. So 25% of the time, one of their children will be 2 big P's at the same time. So 25% homozygous dominant. 50% of the time, you'd expect them to be heterozygous. There's also a 25% chance that they are homozygous recessive. So hopefully this makes sense as to why 75% are purple. Because 75% of them are predicted to get at least one dominant allele, right? These ones, and these ones. And you can use the Punnett square to do the math, right? So that's 3 out of the 4 squares are purple. Right? So that's why it's 3 dominant, 3 purple to every 1 recessive, right? 1 white. So a 3-to-1 phenotypic ratio. So that's what you expect to happen. And again, this one has white flowers because in this case they have 2 recessive alleles, so they can finally express the recessive trait. Then at the bottom, it looks like I want to define something here as we round out Mendelian genetics. I think it's about time to define what that truly means. That means you're talking about a character like flower color in pea plants. And that character is controlled by, I'm going to erase this for more room. Hopefully you've copied it down. A single character like flower color controlled by one gene. That was supposed to say a single gene of which 2 alleles exist in the population. So these are simple genetics. Not all genetics is this straightforward. Because a lot of the time there's not just 2 versions, there's many versions, which complicates things, but it doesn't disprove these principles. It just gets more complicated. So all the 7 pea plant characters that he studied followed the principles of Mendelian genetics. Like for instance, seed color. It's controlled by a single gene of which there's 2 alleles. The allele for yellow seeds, the allele for green. Nothing in between, nothing else, that's it. Okay, so simple. Only 2 alleles exist, 2 options. So now we're ready to go back here. This really summarizes everything. I like this slide. It shows you exactly what happened. This shows you experiment number 2. The Punnett square like we just went through, and they nicely shade the alleles too. Then it shows you the results of the F2 offspring where you can see that 3-to-1 phenotypic ratio, and you can see the variety of genotypes, a 1 to 2-to-1 ratio. Now this is just if you need help more like with the Punnett squares, just to show you that it's just simple math behind it. We already talked about this, so I'm not going to repeat it. I said all this. I want to say this at the bottom. Each of these 4 offspring squares is equally likely to happen. They can't all happen at once. Only one can happen for one kid, but each one is an equally likely event. And it's just basic probability that governs genetics, because everything goes back to that coin toss of independent assortment during meiosis, and it's true chance. It's a 50-50 chance. So same rules that govern coin tosses, cards, and dice. I want to reiterate what a Punnett square simulates. So remember we just did our last one, we did a cross between, let me write it out this way actually. The parents were like this. And I said the first thing a Punnett square does is it simulates the production of gametes during meiosis. So what we did is we did that. We took the genotype and we said, hey, half the time that gamete's going to get big P. The other half of the time it could get little p. Either could happen. 50% chance of each. Okay? So the genotypes of egg and sperm don't have two letters. And this should also tie everything together, hopefully. They only have one letter, right? Why is that? Because they're haploid. Because you took that homologous pair and you just picked one member of it. Just one. So you only have one letter now. Right? The parent, before they do meiosis, their cells are diploid. They have two homologous chromosomes. Right? So these are the gametes. So the genotype of a gamete is always just a single letter. So hopefully that makes sense. One more example. If you are homozygous dominant, big P, when you do meiosis, your gametes, right, your egg or sperm are always going to be big P. 100% of the time, because that's all you're going to get if you pick one of the two. Now, this is really cool. This ties in chapter 10 and what we know in the late 20th century and the 21st century about biology. We're going to answer this question. Well, I'm not going to write it, I'll just say it. Why are white flowers white and purple flowers purple? Why is a heterozygous plant purple even though it has one recessive and one dominant? What's really going on? And to know what's really going on, you have to know something about chapter 10 and gene expression. Number one, you have to know that sexually reproducing organisms are diploid. They have two copies of each gene, one from the dad, one from the mom. Now this picture, it's really cool, is a picture for a heterozygous pea plant. They have one dominant allele and one recessive. But like we said, they're still purple, right? So why? Well, the answer is, if you look at this, here's the big P allele on the top, right? That allele codes for a protein, in this case an enzyme, and the job of that enzyme is to deposit purple coloration, purple pigment, a purple molecule, into the petals of the flower. Okay? So if you have this enzyme, it's going to do its thing. It's going to deposit purple coloration into the petals. And this individual, right, they have one version, one allele that codes for this enzyme. It works. The enzyme's the right shape. But they also have a recessive allele, lowercase p. Remember we said alleles are just slightly different sequences? Well, they're really slightly differences in this case. It's just a one-letter difference. One has a C, the other has an A at that spot. But this example is a recessive allele that is a mutant allele, okay? So this mutation here, that change to an A, was a big problem for the instructions of building that enzyme. Such a catastrophic mutation that you completely disrupt the information. It doesn't even know how to build the enzyme. So, no enzyme. But this plant can compensate for that. It has one working copy of the gene, one broken. That's okay. It can just go to the working copy, express the gene, build the enzyme, has purple flowers. Okay? So that's kind of what's going on, Now, if you look at little p plant. The reason that it has white flowers in that case is it doesn't have any of this. For this individual, both genes are broken, so that individual has no enzyme at all. That plant that's little p has the complete inability to deposit pigment. What does the lack of purple pigment look like? The lack of color? White. And that's why they're white flowers. So, hopefully that was kind of like, not mind-blowing, but kind of tied everything together. And what do we have here? Just putting into writing exactly what I just said over the last 3 minutes. Importantly, our recessive allele was due to a mutation, so our protein's not working, it's dysfunctional. So the enzyme can't do its job. Here, for this slide, don't get intimidated by this. I have all this craziness. I usually do cover this in face-to-face, but I'm not going to here in lecture. All I want you to know is Mendel did more experiments than this. He also tracked 2 pea plant characters simultaneously. Like, for instance, the shape of a seed and the color of a seed at the same time. And I'll summarize it, really straightforward. What he learned is that the inheritance of one character does not affect the inheritance of other characters. So just because you have yellow seeds, which is dominant, doesn't mean you're more likely to have round seeds. You could be yellow and wrinkled, dominant for one, recessive for the other. Okay? So people, animals are not dominant of all their characters at once or recessive all at once. You're mixtures. You can be dominant for one, recessive for another. So the inheritance of one character doesn't affect the inheritance of another. Side note, unless those 2 genes are on the same chromosome, then they might be linked. But don't worry about that unless you're going to be a biology major. Here is just my slide defining Mendelian genetics, which I already did for you in my last picture. And I want to end this by saying the patterns of genetics and inheritance for characters are not always as simple as everything that we've covered so far. The relationship between your genotype and what you actually see, the phenotype, is rarely so straightforward. We'll talk about why that is in our last video for the second half of chapter 9. Mendel published and presented his work in 1865 and 1866, but no one recognized his work while he was alive, probably because he was going around talking about things called invisible heritable factors, and that probably just sounded kind of iffy from the start. Also, he was giving his talk at the same time, I believe, that Charles Darwin was talking to the Royal Society in London about evolution. So he was not, recognized during his time, but definitely one of the key figures in biology.